Okay, are there any questions? If not, let's go ahead and get going. Let me tell you that I graded a whole bunch of labs. So take a look at your labs. You have until Saturday to make updates on the labs that I just graded. Any question about any of that? I don't recall which labs I graded, so if anyone can mention it, please do so. Let me log in.
I
maybe.
think you
got it
graded
to
up till lab 6. Oh, I think someone said lab 7. Never mind.
Go ahead.
I got 7.
So was it 5, 6, and 7, or 4, 5, and 6, and 7? Like I said, I don't remember. I'll have to look. I also did the unknown project, and some of you haven't gotten started on your unknown project. If you don't get started right away, you will not have time to finish your unknown project. So if you need a little bit of help on the unknown project, you can contact me. I sent out 3 tests in your unknown number. I think I sent that all on the same day. And that was a long time ago. So check your Canvas email. I sent you the gram stain, gelatin hydrolysis, and PR lactase test. Or lactose test, one of the two. All right, let me see what labs I did. You're not seeing the screen, correct?
No,
Correct?
we're not seeing your screen.
I'm going to show the grade, so I'm making sure I'm not showing anything to you guys. I'm doing this to this easy way to do it. Let's see, I got quiz 2A graded. I've not graded quiz 3A yet. Quiz 4A. I have to look at that. I'm not sure. I thought quiz 4 didn't have an A, but maybe it does. And I got the couple of worksheet lectures done this week, although that wasn't the last 2 days. I think that was on Sunday and Monday. I think I got worksheet for lecture 5 and the worksheet for lecture 6 graded. So
Dr.
take a look
Volk?
at your notes, but I have graded. I'm in the lab now. Yeah, lab 7 has been graded, lab 6, lab 5. Let me check on that.
Dr. Volk?
Just a second. Yeah, you can still work on lab 5 and all the way to 7. And let me check on lab 4. Yeah, lab 4 was graded also. So you have until this Saturday to make corrections to lab 4, 5, 6, and 7. All right, what's your question?
I submitted an unknown test 2 days ago and I still haven't heard back.
What was your test?
I'll have to look and see just a second.
Because the test has to incubate before I'll send out the results. So if your test is requiring 2 days of incubation, I'll send it out tonight, probably after the lab.
The
And,
ureasis test?
Say again?
Ureasis test?
Urease?
Yeah.
Yeah, that takes 2 days to incubate. So I should get that
Okay.
out tonight if you requested it 2 days ago.
Okay.
But if you
Okay, thank
requested
you.
it one day, you're not getting it tonight.
No, I'm right at 2 days.
Okay, well then you'll get it
Thank you.
tonight. Yeah, everything has to incubate before you get the results. And the long one is MR. If you request the MR test, it has to incubate for 5 days. Gelatin hydrolysis is even longer. That's why I gave you that one at the start, because that takes a week to incubate. All right, any questions on what we've done, what we just discussed? If not, I'm gonna start shutting this down. And let me go ahead and share the screen now. Like I said, if you need help on the unknown project, email me and I'll try and get you started. If you don't get started on that right away, you won't have time to get it finished because that project takes time.
I actually have a quick question about the lab,
Okay,
the latest one. With the sim test, the deep one, if it's if there is a black precipitate, can that interfere with your ability to tell if it's
okay, I'm gonna ask you to come and get a little closer to the computer. I'm gonna turn up the volume. I did not hear all of that. That's partly my fault. I got the volume way down. There, try it again.
with the sim agar deep test?
Huh?
If it does, if there is a black precipitate, that means it's positive for sulfide production. But could that interfere with like your ability to be able to visualize if there's motility or not?
It shouldn't interfere with your ability to determine motility. You can't see the yellow if it's black in the butt, but if it's black, you can just assume it's yellow because you only have hydrogen sulfide production when fermentation is going on.
Okay,
Okay.
thank you.
All right. All right. Are you seeing the screen?
No.
No.
All right. Let me show the screen. There we go. So today is August 12th, and we're supposed to be on chapter 8, which I think we are on. And please understand that you're going to have another quiz this Wednesday. Is that right? Well, today's not.
We had the test yesterday.
I already had the test yesterday. Sorry, I was thinking the 12th. What did I see wrong? Today's Thursday. Okay, that goes to Saturday. I'm just getting confused here. So today is supposedly the last day we're working on chapter 8, and hopefully we'll start chapter 13. I think we're a little bit behind schedule, so we might not make that, but we will try and do that. There is no lab today. I will be there for only 15 minutes. So if you have questions, log in between, what is it, 6:30 and 6:45, is that right? Somebody can confirm if you want. Yeah, I'll be there from 6:30 to 6:45 to answer questions, but there is no lab today. You're supposed to be working on your infectious disease paper and finishing it. It is due this Saturday at 11:59 p.m. and working on your unknown project. All right. So if you have a question about the lab or anything else, make sure you're there from 6:30 to 6:45 because I'm logging off after that point because we're not having a lab. Any questions about what we're doing? All right. If not, I'll go ahead and leave that. And I think I've got there. Hopefully I marked it. My apologies if you're having trouble seeing me. That's not terribly important, but I've got the lights turned off because it's awfully warm in here. I'm just trying to survive.
Do you have air conditioning?
Yeah, but it's not real good in this house.
Uh-oh.
All right, I believe we did discuss chemical mutagens. Any question about any of that? All right. Let's move on. Oops. The frequency of mutation, I've discussed it a little bit, that mutation, spontaneous mutation, obviously always happens whenever the DNA is being replicated. Every once in a while, the DNA polymerase can make a mistake, and it makes a mistake about 1 times in 10 to the 9th replicated bases. Now, because there's about 1,000 bases in your average gene, that means that about 1 in 10 to the 6th replicated genes will be mutated. Any question about that? Now, when we look at the human population, which is, what is it, 7 to 8 billion? Obviously, every day when people are born, somebody's going to be born with a mutated gene, because mutation happens about 1 in every million genes. And I don't know how many births there are in a day, but the population of the planet is between 7 and 8 billion, billion. So that's 1,000 times more than the mutation rate per gene. And let me see if I can remember, there's between 20 and 40,000 genes in the human genome, depending who you ask. Okay, and how you measure it as well. Mutagens increase the mutation rate. You can increase it to 1 in every 10 to the 5th to 10 to the 3rd, meaning 1 in 1,000 replicated genes to 1 in 100,000 replicated genes. This increase in the mutation rate by a factor of 10-fold to 1,000-fold. You can test for mutants in 2 ways. You can have a positive direct selection. And that way you look at the mutant cells because they grow or appear different. Like if anyone were to see this individual walking down the street, you'd say, yep, that's a mutant. That's positive direct selection for determining whether you have a mutant or not. There's also negative indirect selection. And here you detect mutant cells because they do not grow. I've actually done both of these methods for looking for mutant cells. This way I haven't done since an undergraduate, but you can take a colony that has, a plate that has colonies on it, and then get a velvet stamp like this and press the velvet stamp onto the plate. The cells will then be transferred to the velvet. You then take the velvet stamp and you stamp 2 more plates. That puts cells down and the colonies will grow in the same configuration as the original plate. Okay. Well, in the negative selection for a mutation, you're looking for cells that do not grow. And what you're doing is you're plating cells in 2 different media. This media lacks the amino acid, this one lacks the amino acid histidine. And this one has the amino acid histidine in the media. And right here, you can see this colony is growing, but it's not growing in the media lacking the amino acid histidine. So this cells from this colony right here are mutated, and they will only grow in media containing the amino acid histidine. If the amino acid histidine is not present, those cells are mutated and they cannot grow. Any questions about how you check for negative indirect selection of a mutant? All right. Genetic recombination is the exchange of genes between different DNA molecules or different chromosomes. This can happen by crossing over. Crossing over is when physically two DNA molecules come together and then literally cross over. And then one portion, this chromosome has all capital alleles. The other chromosome, let's say it's from the father, all has lowercase alleles. Well, the chromosomes can break and then cross over where this chromosome, which used to be all lowercase alleles, now has some uppercase alleles, meaning this portion of the chromosome crossed over and broke and joined this chromosome. And then this portion here broke off and then joined this chromosome. So a recombination and crossing over, the same thing, slightly different things, or totally different things. Recombination and crossing over. Are they the same, slightly different, or totally different things?
Is it different?
It's slightly different. Recombination is a little more general. Whenever crossing over occurs, you do have recombination. But recombination is every case, including the cases with crossing over, which most of recombination is crossing over. But crossing over is more specific, where the chromosomes come literally together and then exchange parts when they cross over. A recombination is more general whenever a gene changes from one DNA molecule to another. So it's just a little more general. But essentially they are the same. Like whenever crossing over happens, you have recombination. And like I said, when recombination happens, most of the time you have crossing over, but you can have recombination from other events as well. All right. Oh, let me say that recombination does not create new genetic variation within the population. The only way that the population can get new genetic variation is from mutation. Okay? But once the mutation creates the new genetic variation, recombination and crossing over, or just recombination if you want, it shuffles the combination of genes. So you have gene A and gene B, and there could be gene A1 and gene A2, and gene A1 is with gene B. Well, now gene B can be with gene A2. Does that make sense? That's what recombination does. It just shuffles up the genes so they can be expressed in new combinations. Any question about any of that? Like initially, I think the blonde hair gene and the blue eye gene were together, but they recombined, and now you can have brown eye and blue eye genes. All right, genetic transfer. Vertical gene transfer occurs during reproduction. So vertical gene transfer is when the genes of the parents flow to the offspring, or when we're talking about cells, the parent cell divides and the information in the parent cell divides to two daughter cells. Horizontal gene transfer occurs when genes move between cells of the same generation. So one cell gives up the gene, and it may die, it may not. And then that DNA is taken up by a recipient cell. Horizontal gene transfer can happen in 3 different ways. There can be transformation, conjugation, and transduction. Transformation is where DNA comes out of a cell. And the DNA is naked DNA in the environment. That cell probably dies, and then the DNA is released from that cell. The DNA can come up to another cell and be incorporated inside the cell. And then portions of that DNA can recombine with the chromosome of that cell, like the lowercase a gene here, can recombine with the chromosome, and then this cell will get the lowercase a gene. That is recombination, where the cell takes up DNA, brings it into the cell, and then recombines that DNA with its chromosome. So the lowercase a gene can now be incorporated into this cell. This is important. If A is a drug for antibiotic resistance, and this cell is sensitive to that antibiotic, it has now just become resistant to that antibiotic. So the cell has been changed by new DNA that it took up. And this we call transformation. I'm trying to show that there. So we have a little picture, a cell sees DNA, gobbles it up, gets a little indigestion, and then it expresses some of that DNA, getting a new phenotype. Any question about any of that? Conjugation, also called sexual conjugation, is where DNA moves from one cell, this cell, into another cell, this cell. And in gram negatives, let me shrink that a little. In gram negatives, this happens with the sex pilus, that structure that comes out of some cells that have it. And then this cell that has the sex pilus can literally skewer the F- cell. We'll talk a little bit about that. And then this cell can transfer a plasmid, the F plasmid, from this cell to that cell. So this cell gains new genetic material that came from this cell. Okay. Now, gram negatives have the sex pilus, and that's how they engage in sexual conjugation. Gram positives do not have a sex pilus. However, they can engage in sexual conjugation. What happens is 2 gram positives come together, and I don't know what happens to the cell wall. It must get a hole in it. And then the cells fuse their membrane together. And then the cytoplasm of this cell can flow into that cell and vice versa. And a plasmid from this cell can leave this cell and then enter that cell. And this we call a mating bridge in the gram-positive cells. Any question about that? Anyways, that's the 2 ways that conjugation can happen. We'll talk more about it with the sex pilus and the gram negatives. The mating bridge has not been studied as much, so it's not understood as well. But let's just state that conjugation can happen in gram positives by a mating bridge. In the gram negatives, one cell, let's take a look here, is an F+ cell, and it has an F plasmid. And what that plasmid does is it encodes genes for the sex pilus. So this cell has a sex pilus because of a plasmid in this cell that codes for the sex pilus. The sex pilus can literally come out of this cell and then skewer an F- cell. Okay, skewer the cell, and then it can act as a winch, drawing the two cells together. If you don't know what a winch is, it's something you can use like in your car and then pull two cars together. The cells do not have to come physically next to each other because the sex pilus is hollow and the F plasmid from this cell can move through the sex pilus to the F- cell. What happens is the F positive cell will replicate the F plasmid, and then one copy of the F plasmid will move into the F- cell. Now both cells have the F plasmid, and this plasmid codes for the sex pilus. So this cell will now grow a sex pilus, and it will change its phenotype from F- to F+. Any question about any of that? If you don't know why this is called sexual conjugation, ask your significant other. Okay? Any questions about that? The third way that DNA can move from one cell to another cell is with transduction. Transduction uses a virus, and we haven't talked about viruses yet, so let me briefly talk about viruses. Let me blow this up now. A virus can infect a cell, and when it infects a cell, it injects its genome, which in this case is DNA, into its host cell, meaning the bacteria cell. What the virus wants to do is to gain control of the cell, and an easy way to do that is to degrade the host chromosome. Now the host cell's genome can no longer run what's going on in the cell. And then the viral genome runs what's going on in the cell. Any questions about any of that? All right. What the virus does is once it's gained control of the cell, it uses this cell to replicate more viruses. So it will copy its genome and then transcribe its genome to make viral proteins. And then the viral protein will get together with the viral genome to make new virions. And the last thing the virus will do is lyse the cell, and that's to release the viruses from inside the cell. What those viruses do is they then go out in the environment, largely by diffusion, and can infect another neighboring bacteria cell. Okay, and that's just what viruses do. They infect a cell, use that cell to replicate, and lyse the cell, and then they're released. However, viruses make lots of offspring, if offspring is the correct word. And so when they make a mistake, they're not worried about it because they have they have lots of offspring. So if one offspring is wrong, well, it's not too bad. The virus normally incorporates the viral genome into the new virus. Okay, the newly replicated virus So that would be the reddish purple. But you see this cell here? It's incorporated a piece of DNA from this cell here, meaning the bluish purple. And that was a mistake. What happens now when this virus goes on to infect a cell? It will infect the cell and then inject its contents into this cell. But this is not the viral genome. It is some of the genome or some of the DNA from the host bacteria, meaning this partly degraded chromosome. And then that DNA can recombine with the host chromosome. And then this cell can get new DNA. The DNA right here came from this cell right there. This is the third way that a cell can obtain DNA from another cell, and that's by transduction. Transduction happens in bacteria all the time. And in our nose cells, transduction probably happens all the time in our nose cells. However, that doesn't get into the human genome. So nobody cares that, I don't know, my nose cell has DNA from another person in it. Any question about that? The point is transduction probably does happen in humans. It's just that rarely, very rarely would happen to a germline cell. And if it doesn't happen to a germline cell, nobody's interested in it, okay? But in bacteria, transduction happens all the time. Any question about any of that? How often is all the time? Well, actually, let's go back. I mentioned that horizontal gene transfer can happen in 3 different ways: transformation, conjugation, and transduction. In horizontal gene transfer, genes from one cell leave that cell and enter a second cell. And this is one way that genes can move around in a population of cells. For horizontal gene transfer to occur, there has to have been mutation to create the new genetic variation to begin with. But once it's created in one cell, that new genetic variation can spread in the population of bacteria cells by transformation, conjugation, or transduction. Any questions about any of that? Come on. Well, how often do, oh, I got the wrong one here, that's all right. I can't get rid of it. What the heck? Close that down. There we go. There we go. So spontaneous mutation is the only way that new genetic variation can come about. And how often does this happen? About once in every, I don't know if I can do this, 1 times 10 to the 6th genes. Oh, shoot, lost it. Not seeing it there. There it is. So it doesn't happen very often, but it does happen. Any question about any of that? And then transformation can spread around the new genetic variation. Oops. And how often does it happen? About once in 10 to the 5th times, or 10 times more frequently than spontaneous mutation. Any question about that? And the same with conjugation and transduction. Conjugation happens about once in every 10 to the 5th cells, and the same in transduction. 1 in 10 to the 5th cells. So transformation, conjugation, and transduction are all happening 10 times more frequently than spontaneous mutation. Does everybody understand that?
Yes.
Okay. So when you have a gene in the hospital that's a new gene, which most cases it won't be a new gene, and it's spreading around, it's 10 times more likely to spread by transformation, conjugation, or transduction than it is to occur by spontaneous mutation. And we're talking about like a gene for drug resistance or something like that. Any question about any of that? So that's an important point, and that is you need spontaneous mutation to create the new genetic variation. But once it's created, it can easily spread by transformation, conjugation, or transduction. Or I should say more easily spread by those than by spontaneous mutation. All right. Some of the last topics I want to talk about are some specifics about cells. Plasmids are important in cells because a cell is much more likely to exchange a plasmid than it is going to exchange its chromosome. And so a plasmid is much more likely to move from one cell into another cell than a portion of the DNA from a chromosome. Plasmids are very similar to chromosomes. In bacteria, plasmids are small, circular pieces of DNA. They're extra chromosomal because they're not part of the chromosome. But they're like a bacterial DNA in that sense, in that they're circular. They contain DNA, protein, etc. The difference is the plasmid is very small. There's only a few genes, a handful, maybe a dozen or 2 dozen at most on a plasmid. And then the chromosome will have thousands of genes on them. Human chromosomes, which we've got 46 of them, have between 20 to 40,000 genes, okay? I used to know how many E. coli I had, but I'm sorry, I don't remember what it is. Something like 2 to 5,000, something like that. I used to know, but I don't remember. You should also understand that our plasmid replicates independently of the cell's chromosome. And we actually saw that up here with the F plasmid. But this plasmid replicated independently of the chromosome. And then one copy of the plasmid was sent into the new cell by sexual conjugation. All right, any questions about plasmids? You can find them in bacteria. They're not always there. Only some cells have a plasmid. Even some eukaryotes have plasmids, like yeast cells have plasmids. But human cells do not have plasmids. There are different categories of plasmids. You don't need to know the names. All you need to know is that there are different categories or types of plasmids. All right, any question about plasmids and horizontal gene transfer? If not, let's move on to transposons and insertion sequences. A transposon is a piece of DNA that has an inverted repeat, which is called an insertion sequence, because this inverted repeat allows this gene to open up DNA elsewhere and then insert itself in that location. A transposon is a piece of DNA that moves from one location on the chromosome to another location on the chromosome. Everyone get that? So a transposon is a jumping gene, a gene that moves around from one place to another. They're important because the genes are not static on the chromosome. They can move around in a transposon, although they have to get inside a transposon. And they can also move from the chromosome to a plasmid via a transposon. All transposons contain the transposase gene and the inverted repeats. The transposase gene cuts out this portion of the DNA from the host chromosome, and then it will insert in another location, and it'll cut that DNA so it can insert there, and the inverted repeats help it move out and insert. Okay. So all transposons have the inverted repeats, which are the insertion sequence, or IS1, and a transposase gene. A complex transposons carry other genes. Usually they have another gene, like kanamycin resistance, in between two transposons, meaning you have the normal transposon over here and another transposon over here. And then that jumps around the whole segment from one place on the chromosome to another place on the chromosome. So you can carry genes around on a transposon. Why they're important is because, well, it aids in shuffling the DNA. The genes can move from one location on the chromosome to another. They can move from a position on the chromosome to a plasmid. And then if they're on a plasmid, the plasmid is more likely to go into another cell. And so transposons aid the genetic information from flowing from one cell to another cell. Any question about any of that? Here's a transposon, which has moved into a plasmid, and it's carried the kanamycin resistance gene with it. And now if this cell exchanges plasmids, the other cell will gain the kanamycin resistance gene. All right, so hopefully that's explained why plasmids and transposons are important in the flow of genetic information in the population of cells. I can tell you that eukaryotic cells do have transposons, and I know humans have them, but I don't know anything about them. I know that corn has transposons, and transposons were actually first discovered in corn because with Indian corn, which has different colors, people were noticing that the color was moving around, and the color gene, and that the color on the corn cob was also changing. And they studied it and found out it was from a transposon moving the color gene of corn around on the chromosome. All right, genes and evolution. Genetic variation, if it occurs in a population, for all new genetic variation to occur, it has to come from mutation. But once mutation brings it into the population, crossing over can reshuffle it and express that new variant with another gene. Transformation, conjugation, transduction, transposition can all shuffle that new genetic variation around and then exchange it and bring it into new cells. Now, all this genetic variation drives evolution of the population, which occurs via natural selection. Any questions about that? All right. If not, that's it for chapter 8. I guess I should say that. Ah. And I had to shut my syllabus down because it wasn't displaying correctly. I think we're doing chapter 13 next. Somebody might be able to tell me.
Yes.
It could be chapter 14. Chapter 13?
Yes.
Yep, viruses, viroids, and prions. I'm not using my headphone today, so hopefully you're hearing me okay. I'm actually
Yeah,
doing this through
good.
my thumb drive. Say again? I'm good?
Yeah, we can hear you good.
Okay, good. All right, viruses, viroids, and prions. Obviously know the terms. What is a capsid, an envelope? What's the spike? What is a bacteriophage? Know about the lytic life cycle and the lysogenic life cycle. Know what a prophage is, a provirus. In animal cells, know what a latent virus infection is or latent life cycle. Know what a persistent virus life cycle is, a persistent infection. Know what a prion is and know what viroids are. Understand the various characteristics of viruses, their structure and classification. Understand about how viruses multiply. And then lastly, understand about prions and viroids. Any question about what we're doing? The only goal a virus has is to replicate. And how they replicate is they infect a host cell, invade it, hijack the host cell, and then force it to make viral copies, which then escape the host cell. Let's watch a little video on that. Taking its time. Macrophage is a bacteriophage. We'll talk about that.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
All right, any questions? Some general characteristics of viruses. Viruses are inert when they're not within their host cell. They display little or no metabolic activity outside of their host cell. We require a living host cell to multiply. So we can say viruses are obligate intracellular parasites, meaning they're a parasite that requires a cell to live within. Now, whether viruses are alive or not is another question. We're not going to address that issue because people argue over it. Viruses multiply within living cells by using the host cell's enzymes, nucleic acids, amino acids, and other molecules. The virus has no ATP generating mechanism. They need to get their ATP from their host cell. Viruses do have a genome, but that genome can be either DNA or RNA. It is never both. Viruses, besides having the central genome, which is either DNA or RNA, they have a protein coat surrounding their central genome. Some viruses, and this virus is cut out on this edge so you can see inside it. Some viruses have an outer envelope that surrounds the protein coat. The protein coat is usually called a capsid, and then the lipid layer outside of the protein coat, if this virus has it, is called an envelope. Any question about any of that? Viruses have a host range, the range of cells that they can infect. So a certain virus can only infect certain cells, and this we call the host range. The host species are the cells of the species that the virus can infect. This is determined by specific host attachment sites, like some molecule on the cell wall or the flagella that the virus attaches to, as well as maybe cellular factors necessary for viral multiplication. Most viruses only infect the cells of one host species. For example, HIV mainly infects T cells and what is it? White blood cells, or brain cells, one of those, of humans. So HIV mainly infects human cells. It is true that viruses can infect related cells, like HIV can infect one of the chimpanzees. However, if HIV infects the chimpanzee, it does not give the chimpanzee AIDS, meaning that the HIV does not destroy the chimpanzee's immune system. Any question about any of that? The host range is determined by factors that we'll call receptors on the host cell that the virus attaches to. The cell has to have a receptor for the virus to attach there, as well as on the molecules of the virus which attach to the host receptor. And that's why most viruses are specific for attaching to the cells of one or a few species. Now, that's not true of all viruses. As you know, influenza can infect many different animal cells, like horse and pig, human. Rarely, influenza can even attack or infect cats and dogs. Usually, human influenza does not affect cats and dogs. But it does affect horses and pigs. And another one, which is even much different, is that influenza can grow in birds. And so there are some viruses which infect more than one species, but most viruses only infect the cells of one or very few species. Any question about any of that? You should know that viruses are relatively small. Here we have a little bit of a eukaryotic red blood cell. That's why it's red. And there's an E. coli, which the length is actually a little bit larger than your average bacteria. The width is not larger, but the length is. And there we can see even the biggest virus, the longest, is smaller than E. coli. Viruses, their typical range in the diameter is between 20 nanometers in diameter to 440 nanometers. Their length, they can be longer, but that isn't the biggest diameter of the Ebola virus. It's the longest virus, but the biggest is the megavirus by diameter. And you can get them approaching the size of a bacteria cell. 440 nanometers is about the size of the smallest bacteria. But generally, they're about 10 times smaller than your average bacteria, which is about 10 times smaller than your average eukaryotic cell. The smallest virus that you're familiar with is the polio virus, around 30 nanometers. You can get something smaller, but I'm sure nobody's heard of bacteriophage MS2. Okay. Any question about any of that? A new virus is called the virion. So a virion is a single mature, complete infectious virus particle. Virions will have a nucleic acid as its genome, but its genome can be either DNA or RNA. It's never both DNA and RNA. It's one or the other. And that's unusual because all other cells of the domains, the new genome is always DNA. Okay, for the viruses, the genome can also be RNA. The genome can be double-stranded or single-stranded. Double-stranded DNA is normal. That's what our cells have for their genome. Single-stranded DNA, some viruses have a single-stranded DNA genome. That's not unusual. Our cells are single-stranded whenever they are replicating the region of DNA or they're transcribing the region of DNA. Our viral genomes can also be double-stranded RNA, and that's unheard of in our cells. Our cells do not have double-stranded RNA. And the viral genome can also be single-stranded RNA, which that's normal. We do see that in our cells. However, when we're talking about viral genomes, we have to mention whether the single-stranded RNA is the positive strand, single-strand RNA, or the negative single-strand RNA. The positive single-strand RNA genome can be read by the ribosomes. The negative single-strand RNA genome cannot be read by the ribosomes because this will be the complement to the positive strand. Any question about any of that? With viruses, the genome can be linear or circular. It just depends on the virus. Some viruses, like influenza, have their viral genome in several segments. This should not be unfamiliar to you because the human genome is in several segments, and we call them 46 chromosomes. So all viruses have the central genome, and it's surrounded by protein that we call the capsid, the protein coat surrounding the genome. It is composed of capsomeres. We blow this up. So we come over here. Each of these little circles is a capsomere. And in this virus, there's only one capsomere making the capsid or the protein code of the virus. However, that doesn't have to be the case. You can have a virus made up of one or more capsomeres. Whenever you have a viral tail, there will be more than one capsomere because the head will have a capsomere and the tail will be another capsomere. If the virus has no envelope around it, we call this a naked virus. All it has is the viral genome and then a protein coat surrounding it. If it has a lipid bilayer around the protein coat, we call that an envelope. And it is a lipid bilayer. It does have proteins and carbohydrates in it. And we'll talk a little bit more about the envelope when we're talking about animal cells, because they can have an envelope. They can be a naked virus. For most, if not all, bacterial viruses, they don't have envelopes. The envelope is external to the capsid. It's found only in some viruses. Here we're seeing, oops, went too far. A virus with an envelope, and the bluish region is the envelope. You can see the capsid coat underneath the envelope right there. And the capsid coat can look like a dice, a polyhedron dice. And there we can see the viral genome inside the capsid. If the virus has an envelope, it may contain spikes. These spikes are glycoproteins. If the virus has spikes, they are the outermost region of the virus, and it'll be the spikes which attach to the cell that this virus will infect. So if we're talking about a human influenza virus, it's the spikes that attach to our cells. And it's always the outermost protein of the virus which attaches to its host cell. If the virus lacks spikes and envelopes, the outermost layer of the virus is the protein coat, and the protein coat will have the molecules needed by this virus for the virus to bind to its host cell. The spikes, being the furthest out, are used by the virus to attach to its host cell. The spikes are also antigens because they're the furthest out. And antigens are molecules that an animal cell can recognize as foreign to itself and then mount an immune response against. And so the spikes are usually antigens for us. We mount an immune attack against a virus that has spikes. We mount the immune attacks against the spikes because the body can recognize the spikes as being foreign to them, and that's what we call an antigen. All right, any question about any of that? Let me finish talking about the general morphology of viruses. Viruses can be classified on the basis of their capsid structure. You can have a helical virus where the virus is in a helix, like right here, or you can have a polyhedral virus where the protein code is many-sided, like a dice. That could be 6, 8, 20-sided. And then the third viral morphology is a complex virus, where the virus can take on many different shapes. So there's a complex virus, there's a complex virus. That's actually an electron microscopic image of this virus. And there's another complex virus containing an envelope. All right, any question about any of this? We'll talk about bacteriophage T4 next time. Bacteriophage T4 is the virus which has been studied the most by all virologists. All right, any questions? All right, if there's no questions, I'm going to stop here. And I will be in the lab from 6:30 to 6:45. If you have questions, come to the lab at that time. Otherwise, you do not need to come to the lab. All right, I'll see you later.
Thank you.
All right.